Okay, so we're going to be doing the 2020 practice FRQ exam number 1. And as you can see, it'll be number 1. And so a tip/trick that I have is that I tend to read the questions first so that when I start to read the prompt, I know what to look out for. So number 1 says, describe the factors that influence an individual's phenotype. Phenotype we know is the physical characteristics of something. B says, explain how the presence of N. See, we don't know that, but we're not going to stress about it. We're just going to call it N. Chemical cues affects gene expression in the Daphnia. And then C is this one. It's long, so we'll get back to that. And then D says, provide reasoning to justify your prediction in part C. So we know that these two go hand in hand. Alright, so back to the prompt. So right away we see that there's a lot of italicized words. So I'm assuming those are names and that we have to remember them. So for instance, the very first word, since we already underlined it within our questions, we know that we do have to pay attention to it and that it is important. So Daphnia L is a freshwater crustacean that can detect chemical cues produced by the predator and glauca. Researchers raised Daphnia clones and observed that those exposed to N chemical cues during their development produce larger protective crests. So now I feel like this is something we should know. So it says that these that were exposed to N ended up having larger protective crests. So, and apparently that's what figure 1 shows. So this is figure 1. So that's good to know. All right. And as described in figure 1 and 2, crest height and width are measured as ratios to body length. The increase in the size of the crest, width, and height is not associated with a corresponding change in the Daphnia's genome. Researchers hypothesize that growing a crest is energetically costly, so Daphnia do not develop the large crest in the absence of N. So that was a lot of information that eventually we're going to have to go back to truly understand it. But okay. And then here in the image, we can see that they pointed out the width for us. They pointed out the body length for us. You know what? It'll be easier to do that. And then here is again, like you can see it in the description. And also based off what we read, is that this is number 1, which is like the original, like this is what they normally look like. And then this is 2, which is, that's how bigger they get and whatnot after they've developed. As you can see, by the way, the crest width got larger over here. And then the crest height is definitely larger. See? So now we're going to go to the questions. Oh, this is figure 2. We can't forget it. It's showing us the relative trait length in percentages. So this is our Y, our dependent variable. Wait, let me see. This is our Y. And then our X's are the crest height and the crest width. And so the graded area is the chemical cues, and then the white shaded area is the control group. So the control is just like the regular Daphnia. And then the gray shaded is after it's developed through the chemical cues. So again, okay. So again, here we have like the little description that tells you everything. Okay, so A, describe the factors that influence an individual's phenotype. So right away, we know we can start writing about how phenotype has to do with the physical characteristics. I don't think I can write. No, I'll put a text box. So, physical characteristics. One of the things that we can talk about is that the factors that influence an individual's phenotype include genetic information, also environmental influences. And I feel like that would be enough to answer the question in my perspective. So it says describe the factors, so I guess you could talk more about the genetic information. Like, for instance, in figure 1, it is like the crest height and width are significantly larger than beforehand. And then in figure 2, you have like the numbers. So the control for height is like about 10, I'd say. And then after going through the chemical cues, the crest height is like around 40, maybe like 38. So that's almost 4 times larger than beforehand. So I guess you could describe that for the phenotype. And that's really about it. I personally wouldn't know how to bring in environmental into this, because it doesn't, the only environmental thing that it mentions is like fresh water, but then that's about it. So I don't know. I don't know about that one. But now let's move on to B. Hold on. So it says, explain how the presence of N chemical cues affect gene expression in the Daphnia. So I guess the way you can go about this is that you can talk about how the N chemical cues enhance expression of genes associated with the crest development in Daphnia. So that's basically what we already talked about in part A, which includes both figure 1 and figure 2. So it's the same thing, in my opinion. In my opinion, about how the crest width and height are significantly larger, and it did not affect the body length, because based on figure 1, they look exactly the same. And we just highlight that. Okay, so the body length is about the same. That hasn't really changed at all. And they also did not mention it in the question. Another thing you can say is right here, when we underlined that their development produced larger protective crests. So yes, so because they had to go through the N chemical cues, they developed larger and more protective crests. So that's how I would answer part B. For part C, it says, as a follow-up experiment, researchers placed the Daphnia that were exposed to the Notorecta chemical cues into a tank without chemical cues. The Daphnia reproduced asexually, and the offspring developed in the tank without chemical cues. Okay, so before we go on, I would like to point out how it says they produce asexually. So that means that they have the exact same genetic composition, I believe. So the offspring is exactly like the parent. So that's one thing I have to point out. Okay, so it says, predict the relative size of the crest height and width of offspring raised in a tank without chemical cues as compared to the parent Daphnia. Okay, so I basically already answered the question without even knowing, and it's basically that the offspring should look, if not exactly like the parent, then it should definitely resemble the parent, because asexual reproduction is a type of reproduction that does not involve the fusion of gametes or a change in chromosomes. So essentially, the parent will give birth to itself. It'll look almost identical. So yes, yeah, that's how I would answer this question. And width of the offspring would be identical to the parent, and that's basically how it ended. Lastly, part D says, provide reasoning to justify your prediction. Part C. So this shouldn't be that hard, since we basically already explained it in part C. And what I would do is re-describe the asexual part. And then we could say that the reasoning involves that the N chemical cues only affected the phenotype of the Daphnia parents during their development, leaving their genotype unchanged. So that's definitely one thing that we can put down. And then another thing is that since there was no change in the heritable genetic sequence of the parents, the offsprings are not expected to exhibit the larger crest and width like we saw in figure 1 and 2, or that was also described within the within this portion. Let me see. Okay. Within this portion, we would not see that in the predator-free environment. So overall, I feel like we would do pretty good on practice number one. Number one, which is this. And yeah, now I have to do the other FRQ. So in the directions, you mentioned we had to do 2 FRQs. So the second one I'm choosing is Unit 2 Progress Check number 2. And it's because I realized that there isn't really a prompt like the first one. So that's one good thing to note, is that there is no prompt. There's just the directions. So read each question carefully, write your response in the space provided for each part of each question. Answer must be written out in paragraph form. So, okay, that is important to note, is that it must be a paragraph, not just 1 or 2 sentences. And it says, outlines, bulleted lists, or diagrams alone are not acceptable and will not be scored. Okay, so that's great. So figure 1 represents a plasma membrane and molecules present on either side. So, oh no. Okay, so we see the labels. We see the membrane channel, the specific water-soluble ions, the phospholipid bilayer, which, by the way, we know that these are the heads, these are the tails. One is hydrophobic, one is hydrophilic. And then we have the small lipid-soluble molecules. And then, all right, that's about it. Okay. So for A, it says describe, in all bold, the biological need for cells to be surrounded by a membrane that is selectively permeable for different materials. So based on the millions of thousands of times that we have reviewed this, I would say that having a semi-permeable membrane, that's my tip, is to restate the question. So having a semi-permeable membrane allows cells to control the passage of material coming in and going out. This is because we know that semi-permeable membranes only allow certain things to go in. They're not completely permeable, they're semi. And then I would continue to say that it allows cells to maintain homeostatic condition by only allowing certain molecules, again, that's a key word, certain molecules, not all molecules, to cross the cell, which is necessary. I don't know how to spell necessary. Okay, which is necessary for cell functioning and survival. So, for instance, we can see those specific water-soluble ions going through the membrane channel. But that's really all I would put for part A. Now for part B, it says explain how the model shows selective permeability of the membrane to specific ions. So that's kind of funny. That's exactly what I just said, the specific water-soluble ions going through the membrane channel. But I would say that the model shows selective permeability of the membrane to specific ions, since we are shown 2 types of specific water-soluble ions, solid and spotted. As you can see, some of them are spotted and some of them are solid. And, but only the solid colored ions cross the membrane channel. So yeah, that is something to note. And then I would say that the dotted ions do not cross since a membrane is selectively permeable. And in this case, I guess the membrane channel is only allowing the solid ones to pass by, even though, like, evidently both of them are, like, whether they're dotted or solid, they're both water soluble ions. But here we can see that the membrane channel is working hard and only allowing the solid ones. So that would be my answer for B. For oh no. Okay. For C, it says describe the characteristics of the phospholipid bilayer that permit small hydrophilic, see, that's something, small hydrophilic lipid molecules to pass directly across the membrane. So as I mentioned before, this is a phospholipid bilayer. Let me highlight it. Whoa. Okay, that's the phospholipid bilayer. And that has both hydrophilic and hydrophobic areas. And so for part C, since it says describe, I would say how the phospholipid bilayer each have a hydrophilic head and two hydrophobic tails. You see how it has like two, like 1, 2, and then here's the head. Okay, so I would say, I would mention the head and the tail, and how the hydrophobic tails are in the interior of the membrane, and the hydrophilic heads point outwards. And then I would say how the hydrophobic molecules that are small enough can easily pass through the plasma membrane, as you can see here. They can just kind of pass through. Because they are hydrophobic, like the interior of the membrane. And that molecules that are hydrophilic, on the other hand, cannot pass through the membrane on their own because they are hydrophilic, like the exterior of the membrane. And so therefore, they are excluded from the interior because they just can't go through. This area would be off limits for any of the other ones. And then lastly, for part D, it says, based on the model, explain whether the molecules shown crossing the membrane require energy to do so. Okay, so we're going to go back up, look at the model, and I, kind of look at it a little bit closer. And I would put that the specific water-soluble ions, which are labeled as these, I'm going to use a different color, which these are labeled, that cross the membrane channel down here, require energy since they are traveling through active transport. There is a higher concentration of the specific water-soluble ions inside the cell than outside of it. And so then I would continue on to say that the ions crossing the membrane are traveling across the concentration gradient, making it active transport. And then I would say how the small lipid-soluble molecules, on the other hand, travel by passive transport. So those are these that were able to easily pass through this phospholipid bilayer since the interior, is hydrophobic. And so, yes, therefore they do not require any energy, allowing them to easily pass through the plasma membrane. And that would be my answer for part D. Thank you.